Chapter 13, Topic 13.3, Stage 2, Presentation of Antigens. In this topic, the learning objectives are: compare the terms antigen, immunogen, and epitope, list the characteristics of antigens that optimize their immunogenicity, list the types of cells that can act as antigen-presenting cells. In this topic, we're going to talk about the entrance of antigens and antigen processing and presentation. Imogen is a synonym for antigen. Why we need a synonym, I have no idea, but the book refers to Imogen for this section of the textbook. I prefer antigen. I'll be using the term antigen in this recorded lecture on the exam, so just don't get confused with Imogen. Anyway, what is an antigen / Imogen? An antigen is anything that stimulates an immune response. I guess that's where the term immunogen comes from, but antigen is the key for the antibody lock. Whatever helps you remember. Anyway, back to the topic, Teresa. An antigen is anything that stimulates an immune response, but some molecules do a better job than other molecules. Proteins make great antigens because they come in many shapes and sizes, and they tend to be fairly rigid. Think of trying to pick a lock with a cooked spaghetti noodle versus an uncooked noodle. The uncooked noodle will work better, which is why lipids tend not to make good antigens. They are too floppy. Small carbohydrates generally do not make good antigens because, let's be honest, they all look the same. Poor antigens can be made into good antigens by attaching it to another molecule. We call these other molecules either a carrier molecule or haptens. Now, on to what an epitope is. Lots of good antigens have specific areas that a B cell or a T cell will recognize. The part that actually fits into the variable region is the epitope. So a microbe or a pollen grain enters your system. Phagocytic cells gobble down anything displaying a PAMP. In the process of digesting these foreign molecules, bits are displayed on the MHC2 receptors. The phagocytes then wander around contacting T cells. If the antigen on the MHC2 doesn't match the T cell receptor, the phagocyte moves on. If the antigen on the MHC2 matches the T cell receptor, the T cell will begin to divide. So in the next topic, we will talk about what the B and T cells do once they're activated. But for this topic, I want you to remember and be able to compare the terms antigen, immunogen, and epitope, list the characteristics of antigens that optimize their immunogenicity, list the types of cells that can act as antigen-presenting cells. Basically, that's anything that can phagocytize.